Good morning, good afternoon, good evening, AP Bio. My name is Chris Mansour, and I am coming to you live from my room in 102 at Tiffin Columbian High School. Today's topic is 4.1, cell communication, and this is daily video 1. And this lesson will describe how cells communicate with one another over a short distance and over a long distance. And the skill that we're going to focus on is explaining a biological concept or a process. So what will we learn? Always important. So what will we learn? How do cells communicate with other cells over a short distance, but also how do cells communicate with other cells over a long distance? And finally, how is the process of distance communication different from the process of communicating during cell-to-cell contact? So again, you may want to pause the video here, jot down some answers or some ideas you might have. So cells communicate with one another through direct contact with other cells. And so cells of multicellular organisms, like yourself, often maintain physical contact with other cells or make physical contact with other cells during certain activities. Some unicellular organisms live in colonies or in physical contact with other organisms in that colony. So if situations get stressful, they may form a colony to share resources and ensure survival. Cells can also send chemical signals directly into adjacent cells. So if cells are sitting really close to each other, they can send a chemical signal back and forth. And so cell membranes and cell wall modifications also allow for communication to occur between adjacent cells. So in plant cells, they have plasmodesma, which are these structures that allow the plant cells to communicate with each other. And then in animal cells, there are things called gap junctions. So again, there's different structure, different function, but communications evolved in many different organisms in many different ways. And so cells communicate with one another over a short or long distance. So cells use chemical signals to communicate over a short and long distance. So the cell receiving the signal is referred to as the target cell. That's important. That's an important term there, target cell. So when we talk about short distance communication, cells send out local regulators or signals. So a local regulator, think of local as being close. The target cell, again, as I said before, the cell that's being receiving the signal is within a short distance of the signal. So again, local signaling. Often used to communicate with cells of the same type. So they have the same communication. They're in close proximity, so they're going to use the same kind of communication. Long distance is a little different. The target cell's not in the same area as the cell emitting the signal. So if you had to think about a message traveling through your body, if it has to go from, say, your head down into your abdomen or one of those organs there, the cells are not the same. So the cell emitting the signal may be different than the cell receiving the signal. So when those cells signal, the cell signals travels a long distance to reach a target cell. A lot of words that sound the same there. But anyways, often used to signal cells of another type. Hopefully you followed that. But anyways, so let's look at an example. Immune cells communicate through cell-to-cell contact. And so this is B cell activation. So immune cells interact by cell-to-cell contact. So if we look where that arrow just popped up, there's a cell that's infected. It has these little red structures on its surface. We'll call those antigens. And there's our B cell. And if you notice on that B cell, it has a structure that just kind of matches that antigen right there. And so B cell activation is triggered by the binding of a specific antigen to the B cell receptor. So they fit together. And as you can see, when they fit together, if you follow my arrows around, that B cell will then cause the release of another chemical and so on and so forth. But the key there is cell-to-cell contact. They have to match up. So let's look at an example. So neurotransmitters communicate using local regulators. And so exocytotic vesicles release signals. And so there's one of these signaling molecules, which is a neurotransmitter in this case. The signal will travel a short distance, that space between the two neurons, and it'll go across that gap between the cell. And once it attaches to the target cell, it's going to cause a response. And so there again, where that circle is, you can see the movement of those signals back and forth. And let's do a little practice, because practice makes perfect, right? We're getting you ready for that exam in May. So our lesson skill focus will be concept explanation. So explain biological concepts or processes. So here is a multiple choice question for you. A hydrophilic peptide hormone is produced in the anterior pituitary gland located at the base of the brain. The hormone targets specific cells in many parts of the body. So which of the following best explains, again, that word's in bold, so it's important, a possible mechanism that would enable the hormone to efficiently reach all of the target cells in the body? I am not going to read the options A, B, C, or D to you. So pause the video, write down your answer, get some ideas, and then unpause. All right, and I'm back. So I don't have anybody in my room. I'll pretend I do. So who has A? A couple of you. B, all right, good. C, maybe a couple of you. D. All right. Best answer is C. The hormone is released in the bloodstream where it can be transported to all cells with the correct receptors. If you got it, good job. Give yourself a gold star. If you missed it, you may want to pause the video, go back a couple slides and see where I talked about long distance communication, or again, talk to your teacher or one of your friends on how you got that answer. So what should we take away? The key takeaways are: 1, cells use local regulators to communicate across short distances. 2, cells can release chemical signals with the ability to travel over a long distance to target cells of other types. And 3, structural modifications of the cell membrane and cell wall allow cells to send chemical signals directly to adjacent cells. So again, I want to thank you. I want to thank all those AP biology teachers watching this video. I know these are strange times and we're working hard. To all you AP students, keep working hard. And this is Mr. Mansour signing off from room 102. Have a great day, and I'll see you in a later video. And good afternoon, good evening. My name is Mr. Mansour, and I'm coming to you from room 102 at Tiffin-Clement High School. This is topic 4.2, Introduction to Signal Transduction. And today, this is daily video 1. The lesson will focus on the components now of a signal transduction pathway. The skill will focus on describing a biological concept or process. So again, what will we learn? Always important to have goals, right? So what will we learn today? What is a signal transduction pathway? Why do cells use signal transduction pathways? How does a signal transduction pathway begin? What is the role of protein modification in signal transduction pathways? And finally, what is the role of phosphorylation cascades in signal transduction pathways? So signal transduction pathways link signal reception with cell response. So I have an image here of biosignaling. As you can see, there's 3 steps. And the 3 steps of cell communication are reception, transduction, and response. So reception is detection of a signal molecule coming from outside the cell. And so if you look at reception there, you can see at number 1, there's a molecule attaching to a receptor. Transduction is how we convert that signal to a form that can bring about a cell response. So there's that transduction, which is our second step, and you can kind of see that process there going through the cell. And then response, that is the specific cell response to the signal molecule. So that can be anything from transcription to production of a protein. And so many signal transduction pathways include protein modification and phosphorylation cascades. Again, some new terminology may be here for you, so if you want to pause the video and write them down and maybe look them up to define them to kind of help you. But regulate protein synthesis by turning on or off genes in the nucleus. And so that's one of the ways that there can be protein modification. Can regulate activity of proteins in the cytoplasm depending on the function of it. And also there are cascades of molecular interactions, relay signals from receptors to target molecules. So if you think of a cascade, think of water like cascading over a waterfall, it's going to pick up and it's going to be able to amplify the effect. And so phosphorylation cascades also enhance and amplify the signal, as I said. And so these cascades and phosphorylation cascades are important to get the signal to where it needs to go. So let's talk about signaling. So signaling begins with the recognition of a chemical messenger, a ligand, by a receptor protein and target cell. If you look at my image here in a second, you're going to see where I focus in on the ligand. So the ligand binding domain of a receptor recognizes a specific chemical messenger. The key is specific. They have to fit together, which can be a peptide, a small chemical, or a protein in a specific one-to-one relationship. And it's very important that the ligand has to match the receptor. And then a G protein coupled receptors are an example of a receptor protein in eukaryotes. And I'm going to show you here in a second where that G protein is found. And so here is that hormone is binding to that receptor. And as you look at the structure, they fit together. What I have in a circle here is this is where your gene protein coupled receptors are located. So you have GDP to GTP, which I'll talk about. And so GDP is inactive. Right, so that G protein is inactive when GDP is present. When it becomes GTP, it activates, and as you can see in the image, that GTP is going to move over, and it's going to activate the adenyl cyclase. So I'm learning my words a little bit, and then once that's activated, activates ATP. ATP then will be converted to cAMP, which will then activate a protein kinase. A lot going on there. Like I said, I would redraw this picture, number them 1, 2, and 3 as you review. And so, as we move on again to the signaling cascades, these cascades relay signals from receptors to target cells, often amplifying the incoming signal, so amplifying or increasing it. This can result in the appropriate response by the cell, which can include cell growth, secretion of molecules, or gene expression. Just depends on what the cell has to do. So again, if we look at step 1, there's your non-steroid hormone. It's going to attach to a receptor. There's our GDP at step 2. It's now been activated to GTP. If we go to 3, the GTP then will go over to our next protein, which will then, that protein will then convert the ATP to that C-amp. There's our secondary messenger, and then that C-amp will ultimately lead to an effect on a cell function. Again, a lot going there, so you may want to pause the video, and I really do think if you draw these out, it does help. And so, as I just said, after the ligand binds, the intracellular domain of a receptor protein changes shape, initiating transduction signal. Again, this is a model, so I'm not showing the change in the shape there. And then the secondary messengers, such as the cyclic AMP, are molecules that relay and amplify the intracellular signal. So I wanted to show you the picture first before I gave the explanation. So again, pause the video and mark up your, draw the picture and mark up the picture. And then finally, binding of ligand to ligand gated channels can cause the channel to open or close as well. So there we go. I've just focused in on how the C-amp will cause an effect on cell function. So let's look at an example. So signal transduction pathways. So here's a protein pathway and a steroid pathway. And as you can see, we have hormone and vesicles. Exocytosis occurs, so materials are released out of the cell. If we follow the one on top, which is in blue, that'll attach to a receptor protein that's on the target cell membrane, which will then initiate the secondary messengers in the cytoplasm. So as you follow those sequence of arrows and shapes, that would be your secondary messenger, which eventually, depending on the process, may end up in the nucleus. If we look at the bottom one with the steroid in the red, it can pass right through that target cell membrane. Then there is a receptor in the nucleus. It attaches to that receptor in the nucleus, which then ultimately can make the protein. The big difference between the protein and the steroid one is the protein one has to have a receptor protein on the target cell membrane, where the steroid one can pass right through. Structure determines function. So let's do a little practice. So our lesson focus here will be concept application. So we're going to describe, again, the word is in bold, so that's the key point, describe biological concepts and or processes. So here we go with our multiple choice question. Metformin is a drug used to treat type 2 diabetes by decreasing glucose production in the liver. AMP, activated protein kinase, is a major cellular regulator of glucose metabolism. Metformin activates AMPK in liver cells but cannot cross the plasma membrane. By blocking AMPK with an inhibitor, all right, researchers found that AMPK activation is required for metformin to produce an inhibitory effect on glucose production by liver cells. A lot to read there. Pause the video, maybe read it again, talk amongst your friends or in Zoom if you're on that, but here's the actual question. Which of the following best describes, right, that was our main term there, the components that metformin represents in a signal transduction pathway that regulates glucose production in the liver? So pause the video, look through A, B, C, and D. I'm not going to read them to you. Pause the video, come up with an answer, talk to your friends in Zoom, and then unpause the video. And I'm back. So again, I'm in my room. If I was here, I'd be asking my students what they thought if they were here. So who has A? Anybody? B, C, D? All right. Those that picked B, gold star for the day. It is a ligand that activates the signal transduction pathway of the activation of AMPK. So again, best answer for the description is B. If you got it right, good job. Maybe if you missed it, you may want to go back through and look at some of the other options. So what should we take away? So 1, a signal transduction pathway is the binding of a signaling molecule to a receptor located on the cell surface, which I showed you, or inside the cell that triggers events inside the cell to invoke a response. Again, pause these key takeaways if you're trying to process it all. 2, cells use signal transduction pathways to link signal reception with cell responses, so like growth, secretion, gene expression, whatever has to be accomplished. A signal transduction pathway begins when a receptor or ligand binds to external receptors or an intracellular receptor. Again, depending on what kind of molecule it is, determines if it's going to attach on the cell membrane or inside the cell. The role of protein modification in signal transduction pathway is to cause a conformational shape change due to the ligand binding. The change elicits an intracellular response, which causes the second messenger to be activated. So again, if you're like, what's he talking about? Go back a couple slides, and that drawing that I did, you can see that there. And finally, a phosphorylation cascade is a signaling pathway where one enzyme phosphorylates another, causes an amplification or speeds up the reaction, leading to the phosphorylation of thousands of proteins. So again, I want to thank you for sharing this time with me. Again, shout out to all you AP biology teachers out there. I know this is not an easy time to be doing this. Shout out to all you AP biology students. You keep working hard. And again, this is Mr. Mansour signing off from room 102 at Tiffin Columbian High School. Have a good day, and I'll see you in a future video. Good morning, good afternoon, good evening, AP Biology. My name is Chris Mansour, and I'm coming to you from room 102 at Tiffin Columbian High School, and this is topic 4.3, signal transduction pathways. This is daily video 1, and this lesson will describe the role of environment in eliciting a cellular response. The skill will focus on providing reasoning to justify a claim. So what we learn, again, objectives are important. So what we learn, what role does the environment play in eliciting cellular responses? And how are signal transduction pathways used to influence cell responses when there are changes in the environment? So again, pause the video, maybe try to answer these on your own and see how you did. So signal transduction pathways influence how the cell responds to its environment. So the environment is not static, it's constantly changing. And organisms need to regulate pathways to respond to changes in the environment. I have a typical pathway here on this slide. So epinephrine, which is a signaling molecule, will bind to that red receptor there. When it binds that receptor, so the epinephrine is not going into the cell, it's going to activate that G protein. GTP is active. What you're not seeing on my image is GDP, which is the inactive form. But when GTP comes active, it activates that G protein. Then that G protein will slide over and attach to the adenyl cyclase. Once that adenyl cyclase is activated, it will activate the ATP, which then becomes CAMP. So again, a series of events there. Draw the picture out, maybe annotate it. The key point there is that once that CAMP is activated, that's going to activate protein kinase A. Then that protein kinase A will then lead to the glycogen breakdown. So often in a fight or flight response, if your body is requiring more energy, that glycogen that's stored is going to get broken down to glucose. This is that pathway that would fall. And so your cells are responding to environment, in this case, needing more glucose. So the epinephrine, which when it attaches that receptor, will ultimately lead to that glycogen breakdown, release that glucose, and provide the energy that is needed. So the ability to respond to stimulus is characteristic of life and necessary for survival. So again, this pathway kind of demonstrates that process of what happens when a stimulus occurs and the response. So signal transduction may result in changes in gene expression and cell function as well. So signaling pathways can target gene expression and alter the amount and or type of particular protein produced in a cell. Depending on what is needed, it will determine how much is made. So changes in protein type and or amount can result in a phenotype change as well. And so apoptosis can be the response of a signal transduction, which is programmed cell death. And so again, depending on what is needed, if you need more of a protein, less of a protein, or if cells are being broken down, signal transduction will be responsible for that. So let's look at an example. So using signal transduction response to the environment. So in this case, we have quorum sensing is a signaling mechanism used by bacteria. Bacteria have to talk to each other. And so bacteria release signals to neighboring bacteria. So if you see in this picture, this is biofilm development. And so we've got what's in green there are some bacteria. And so they have to talk to each other to ensure that they're all kind of doing the same thing. And so what quorum sensing does allows bacteria to detect changes in population density. And so what will happen is as enough population, it gets to a threshold, enough of those bacteria are going to be present, gets to a threshold, that they're going to then all do the same response. So basically what quorum sensing does, it allows bacteria to act as a group in response to changes in the environment to their benefit. So let's practice. Lesson skill focus, argumentation. We're going to provide reasoning to justify a claim by connecting evidence to biological theories. Again, reasoning to justify a claim. So here's our argumentation question. Again, it's a multiple choice. During a fight or flight response, epinephrine is released in the body's circulatory system and transported throughout the body. Some cells exhibit a response to the epinephrine while other cells do not. This question should sound kind of familiar. Which of the following justifies the claim that differences in components of cell signaling pathways explain the different responses to epinephrine? Again, a lot of words, maybe they're new, pause the video. And then, because I'm not going to read those responses to you. So go ahead, pause the video, talk to your friends on Zoom, ask your teacher a question, read through A through D, and then unpause when you're ready for the answer. Okay, and we're back. So again, I'm in my classroom. My students are not here right now, so let's pretend that they are. So who has A? All right, hands up. B, C, D. Correct answer is A. Good job. Give yourself a gold star. Cell signaling depends on the ability to detect a signal molecule. Not all cells have receptors for epinephrine. only cells with such receptors are capable of responding. If you don't have that receptor from epinephrine, it won't bind to it, therefore it won't cause those other responses. If you picked B, C, or D, maybe look back through where I discussed epinephrine pathway, or maybe ask your teacher a question or whatnot. But again, it's always good to learn from when we don't necessarily get the correct answer. So what should we take away from today's video? The key takeaways: the environment is not static. Again, it's constantly moving, and organisms need to respond to changes in the environment. If you don't respond, you may not survive. The ability to respond to stimuli is a characteristic of life and necessary for survival. 2, signal transduction pathways are used to influence cellular responses when the environment changes. For example, that fight or flight example I gave you. Transduction pathways can regulate gene expression in response to change in the environment or lead to apoptosis, which is programmed cell death. So again, I want to thank you. Again, I know you're all working hard. Hopefully you're finding these videos helpful. And again, this is Mr. Mansour signing off from room 102 at Tiffin Columbian High School. Have a great day and keep studying. Good morning, good afternoon, good evening, AP Bio. My name is Chris Mansour, and I'm coming to you from room 102 at Tiffin Columbian High School, and this is topic 4.3, signal transduction pathways. This is daily video 2. This lesson will explain how changes in signaling molecule structure affect activity, and the skill will focus on how to predict, key point there, the effects of a change in a system based on a model. So what we learn, again, objectives are always important. So what will we learn? How can mutations in the receptor protein or any component of the signaling pathway affect the transduction of a signal? How can chemicals activate a pathway, and how can chemicals inhibit a pathway? So changes in signal transduction pathways can alter cell response. So let's look at the picture first. So I have a normal cell pathway and I have a mutated cell pathway. So there's our hormone in the vesicle. So if we look at the normal pathway, the blue hormone's released, attaches receptor protein, initiates that secondary messenger system cytoplasm, will then, whatever the response will be. But let's say a mutation occurred, or we changed the structure of that protein that's embedded in the cell, in the target cell membrane. That receptor can no longer fit, or excuse me, that molecule can no longer fit in that receptor protein, change the structure, change the function. So if you notice that if it cannot bind to the receptor protein since it's been mutated, you can no longer have that secondary messenger system in the cytoplasm. You've essentially broke that pathway. And so mutations in any domain of the receptor protein or in any component of the signaling pathway may affect the downstream components by altering the subsequent transduction of the signal, which again is what I've shown you in the image. So change in protein structure can result in change in function, like I just said. And so one disruption in the pathway can affect the downstream reactions. So if that secondary messenger system never occurs, anything after it will not happen. So chemicals that interfere with any component of the signaling pathway may activate or inhibit the pathway. So in this situation, we're going to activate a pathway. So here's FGR proteins. They're inactive. That's an inactive signaling pathway. So those proteins are separated. A fibroblast, growth factor, say that 3 times, is released. And as you notice, when those growth factors are released, they cause those conformational change. We've now activated that signaling pathway. And so in this case, we've activated, whereas in the previous one, we inhibited it. And again, this is just a simplified model of the FGR activation. So there you go, there's the proteins focused, and then there they are activated. And here's another one. So chemicals, again, that can interfere with any component of the signaling pathway may activate or inhibit the pathway. So here's a normal nerve cell. There's our neurotransmitters in red. I'm going to focus on the normal one first. Again, those vesicles release the neurotransmitter into the space in between. As you can see, those nice little red circles will attach to a target cell. They attach those receptors, which are the black structures there, and they'll cause a response. If we go into the inhibited one, there's our nerve cell. There's our neurotransmitters, but in this case now, the neurotransmitter has been blocked. So those green little structures there are blocking the release of that neurotransmitter. We've blocked the release of the neurotransmitter. We've essentially blocked any kind of communication going to that target cell. And so again, there's normal, there's inhibited. So let's practice. Again, we're going to predict. So again, argumentations are less than skill focused, but in this one, we're going to predict. Predict the cause or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So, a lot to read here. I'm going to read it to you. So, the insulin receptor is a transmembrane protein that plays a role in the regulation of glucose homeostasis. The receptor's extracellular domain binds specifically to the peptide hormone insulin. The receptor's intracellular domain interacts with cellular factors. The binding of insulin to the receptor stimulates the signal transduction pathway that results in the subcellular translocation of GLUT4, GLUT4 as I tell my students, a glucose transport protein that is stored in vesicles inside the cell. A simplified model of the insulin receptor signaling pathway is shown in figure 1. So if we look at figure 1, you might want to pause the video, try to look at it and write a caption on your own, but there's what's going on in figure 1. So here's the question. Which of the following statements best predicts, again, predicts is the key here, the effect of a loss of function of the insulin's receptors intracellular domain? Pause the video. Again, I'm not going to read the options to you, A, B, C, or D. So pause the video, try to internalize the question, internalize the image, unpause it when you think you have an answer. All right, and I'm back. So again, we'll pretend my students are here. Who has A? Hands up. Okay. B, C, D. Correct answer is B. Again, if you got that, good job, gold star, whatever. If you missed it, you may want to pause the video here, see why you picked A, B, or D. If you're in a Zoom chat, maybe talk to your teacher, your other friends. But again, use the image to help answer the question. So what should we take away? Key takeaways here. A mutation, change in the DNA that alters the ligand receptor specificity can lead to incompatibility, which can alter the signal transduction pathway. The receptor will not undergo proper conformational changes, resulting in inactive internal pathway. If you go back to that one image where I had the normal and the mutated pathway, that should help you out there. Chemicals can activate a signal transduction pathway, which can lead to amplification of a pathway. Again, I have an example for you a couple slides back. And chemicals can inhibit a signal transduction pathway, which can lead to pathways not occurring. So again, those are key takeaways. I have an image for each one of those. So again, I want to thank you. I've really enjoyed filming these AP dailies. Again, huge shout out to all you AP biology teachers out there. I know you're working hard and you're doing the best for your students, and to your AP bio students, keep working hard for your teachers. You guys are doing great. And this is me signing off, Mr. Mansour from room 102 at Tiffin Columbian High School. Have a great day and have a great rest of the year.
All right, hello everybody. Welcome back to AP Daily. My name is Rachel Lytle and I teach at Brentwood High School near Nashville, Tennessee. And I'm ready to talk to you about topic 4.4, which is feedback. So what will we learn? In this video, we'll look at what feedback mechanisms are, how negative feedback affects homeostasis, and how positive feedback affects homeostasis. So let's get started. Organisms use these feedback mechanisms to maintain their internal environments and respond to environmental changes. So organisms are constantly living in environments that are changing internally and externally. So organisms use the process of homeostasis to maintain a stable internal environment. So if you look at this model, you can kind of think about that as a balancing act. And homeostasis is attempting to maintain some sort of target set point in the organism. For example, maybe it's your body temperature or the pH of your blood that needs to maintain a specific number and a target set point for the organism. Right, these feedback mechanisms are processes that are used to maintain homeostasis by either increasing or decreasing a cellular response to an event. The first type of feedback we'll look at is negative feedback. And these negative feedback mechanisms help to maintain homeostasis for a particular cell condition by regulating certain physiological processes. If a process gets disrupted, the negative feedback mechanism can help to return to the target set point. So if we look back at our model, if we were using a negative feedback mechanism, there would have been some sort of disruption to the system, and the negative feedback mechanism would be able to reset the process back to its target set point. These processes can operate at the molecular and the cellular levels. So next we'll look at positive feedback mechanisms. These work a little differently because they amplify responses and processes in these organisms. So in this case, a variable is going to initiate a response to actually move further away from that target set point, disrupting homeostasis. And then it will amplify that stimulus so that the process continues to occur more and more. So if we look back at our model, we've got our target set point. There's been some sort of disruption, and instead of returning to our target set point, we're going to amplify that signal so that we get further and further away from the target until there's some sort of cell response. So let's look at an example. The example we're going to look at is one of positive feedback, and it has to do with fruit ripening. So in this image, we can see that we have some apples on a tree. And what happens is when one apple ripens, it releases something called ethylene gas. And that ethylene gas can then travel and get detected by other nearby apples on the tree. And those nearby apples then start to ripen themselves and release ethylene themselves, which gets detected by even more apples on the tree to release ethylene themselves and ripen. And so then this process repeats itself and gets amplified because as more apples ripen, more apples release ethylene, and then more apples ripen, and then more apples release ethylene. And so we've got that amplification part of the positive feedback process. So now it's time to practice. In this lesson, we're going to be looking at the skill of argumentation and specifically predicting the cause or effect of a change in or disruption to one or more components of a biological system based on a visual representation. And so in this case, we're predicting the cause or effect of a change in one of these feedback mechanisms. So here is our question. You can go ahead and take a second to pause the video, read the question, and come up with an answer. And then when you're ready, start it and we'll explain what happens. All right, so if you're looking at this question, it is telling you that in our figure, we are looking at a model of erythropoietin and how your body reacts if you're at a higher elevation. So at a high elevation, there is less oxygen available. And the question asks you to predict what the response will be based on the blood oxygen level decreasing in that high elevation. The correct answer to this question is B, that there will be more erythropoietin that will be secreted from the kidneys, increasing the production of erythrocytes. And we know that that's the answer because if we look at our figure 1, we can see that when there's low levels of oxygen in the blood, that will stimulate more erythropoietin secretion from the kidneys. And that also stimulates then more erythrocyte production in the bone marrow, which matches best with choice answer B. So what should we take away from this lesson? Some of the key takeaways are that these feedback mechanisms are processes that help maintain homeostasis by increasing or decreasing a cellular response to an event. Negative feedback mechanisms maintain homeostasis for a particular condition by regulating those physiological processes and helping to return them to their target set point. Positive feedback mechanisms amplify those responses and processes in these biological organisms. Thank you for joining AP Daily today, and I'll see you next time. Hi, welcome back to AP Daily. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 4.5, the cell cycle. So let's get started and look at what we're going to learn today. Today, we will talk about the role of interphase, what happens during interphase, the role of mitosis, what happens during mitosis, and the role of cytokinesis. So the cell cycle is a highly regulated series of events for the growth and reproduction of cells. And it consists of two highly regulated processes, the first of which is interphase, where the cell is going through processes of growth and preparation in order for cell division. After that is M phase, which consists of mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. So let's look a little bit more closely at those three parts that make up interphase. The first of those is G1, where the cell is growing. After that is the S phase, where the DNA is being copied so that there is enough DNA in the cell for it to go through cell division and have a copy for each new cell. The third part of interphase is G2, where the cytoplasmic components are doubled in preparation for cell division, again, so that there is enough for the 2 cells at the end of this process. So in eukaryotic cells that are going through cell division, they are transferring genetic information through these highly regulated processes. And mitosis plays a role in this by being a part of growth, tissue repair, and asexual reproduction. So mitosis's role in growth could be when you start as a single cell and then you become 2 cells and then more cells and then more cells until you are what you are right now, which is trillions of cells. That happened because of mitosis, and that was growth and its role in growth. If you are talking about tissue repair, if you happen to get a paper cut, the process that would occur to fill in that paper cut would be mitosis and cell division. And some asexual reproduction in single-celled organisms is done also by mitosis. Mitosis ensures that there is a complete transfer of the genome from a parent cell to 2 genetically identical daughter cells so that they are exact replicas. And mitosis alternates with interphase in the cell cycle, and they cycle back and forth. Mitosis occurs in a sequential series of steps, starting with prophase, moving to metaphase, then anaphase, and telophase. And lastly, mitosis is followed by cytokinesis. Cytokinesis ensures that there is an equal distribution of cytoplasm to both of the new daughter cells. And there are some cells that can enter what we call G0, and cell division no longer occurs in this phase. A cell can re-enter the division of the cycle with the appropriate signals. Non-dividing cells may exit the cell cycle, or they may be held in a particular stage. So let's practice. Our skill focus for this lesson is using statistics and data analysis. Particularly, we are going to perform the mathematical calculation of percentages. So we have a practice question here where it is asking you to calculate a percentage looking at the data that is in this figure. So go ahead and pause the video here for a second. Try to answer this question on your own and then restart it in a second and we will talk about the answer. All right, so in this question, you're looking at figure 1 where we have a pie chart that is telling us about the time spent in each phase of the cell cycle. And the question asks you, what percent of the time required to complete a full cycle do the cells typically spend in interphase? So we need to know 2 things to answer this. We need to know how much time was spent in interphase, and we know that interphase is G1, S, and G2. So if we add those up, that totals to 19. Then we need to know the time for the full cell cycle, which is all of those phases in the image. So if we add those up, they equal 20. So to find the percentage, we would do the time spent in interphase divided by the time for a full cycle. We would multiply by 100, and we get that it would be 95%. So if you chose answer choice D, you would be correct. So what should we take away from this lesson? Some key takeaways are that interphase's role is to allow newly divided cells the opportunity to grow, maintain normal cell function, and prepare for cell division. During interphase, cells grow, they replicate their DNA, and prepare for cell division. Mitosis can play roles in growth, tissue repair, and asexual reproduction. During mitosis, genetic information is transferred from the parent cells to the daughter cells. And cytokinesis is the process that ensures that the distribution of cytoplasm is equal in the new daughter cells. Thank you so much, and we'll see you next time here on AP Daily. Hi, welcome back to AP Daily. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you a little bit more about topic 4.5, the cell cycle. So what will we learn in this video? we will take a closer look at what occurs during prophase, metaphase, anaphase, telophase, and when cytokinesis occurs. So mitosis is a process that ensures the transfer of a complete genome from a parent cell to daughter cells. So if we look at this model, we can see that we have a parent cell that has 2 chromosomes. And after mitosis, 2 daughter cells are created that have identical copies of that genome and those exact same 2 chromosomes. So the daughter cells carry genomes that are identical to the parent cell's genome. So mitosis occurs in a sequential series of steps, and the first step is prophase. In prophase, the nuclear envelope begins to disappear. As you can see in our image, it has disappeared at this point. The DNA coils into visible chromosomes that look like this X shape in the image. And there are fibers that begin to move the double chromosomes toward the center of the cell for our next phase, which is metaphase. So you can see here that the fibers have aligned the double chromosomes across the center of the cell, and now they are all lined up and ready for our next phase, which is anaphase. In anaphase, those fibers separate the double chromosomes into single chromosomes, or chromatids, that you can see here in the image. Chromosomes are separated at the centromere, which is represented by the white dot, and was the middle of the X before they separated into the single chromosomes. And these single chromosomes, or chromatids, migrate to opposite sides of the cell. So where we started with having just 4 chromosomes in our cell, we now have split those into 8 single chromosomes. After anaphase is the phase of telophase. In telophase, we have the nuclear envelope that reappears and establishes 2 separate nuclei for our 2 new daughter cells that will eventually form. Each nucleus contains a complete genome. We can see that there are 4 chromosomes in each of those nuclei, and we originally started with 4 chromosomes in our parent cell. The chromosomes will begin to uncoil. Cytokinesis is occurring and will separate the cell into 2 daughter cells, each containing identical genomes. So let's practice. Our skill focus for this lesson is representing and describing data. So our goal is to describe data from a table or a graph, including describing trends and/or patterns of the data. So if you look at our practice question, you will see that it includes a graph, and the question asks you to pick the best description of that figure. So take a second here and pause the video and try to answer this on your own. And when you're ready, press play and we'll talk about an explanation. All right, so you can see in figure 1 that they've given you a graph that has each of the stages of cell division and tells you the number of chromosomes per cell. And looking at the graph, we can see that anaphase and telophase both have double the number of chromosomes than the other phases of the cell cycle. So we need to look for the description that correctly identifies why anaphase and telophase have double the number, and the correct answer is choice C. Choice C says that chromosomes enter metaphase containing 2 chromatids attached by a centromere. Those were our doubled chromosomes. During anaphase, the chromatids are separated, each becoming a chromosome, and cytokinesis distributes those into the 2 separate cells. So while we are in anaphase, we have double the number of chromosomes than we did in the other phases. So what should you take away from this video today? Our key takeaways are that DNA becomes visible and the nuclear envelope disappears during prophase. The chromosomes are aligned across the center of the cell during metaphase. Double chromosomes are separated into single chromosomes, or chromatids, that migrate to opposite sides of the cell. DNA uncoils and the nuclear envelopes reappear, and 2 new nuclei form, and each nucleus contains a complete genome in telophase. And cytokinesis begins at the end of mitosis and separates the cell into 2 daughter cells. Thank you for joining AP Daily today, and I will see you next time. Hi there, welcome back to AP Daily. This is Rachel Lytle here to talk to you about topic 4.6, regulation of the cell cycle. So let's get started. What will we learn in this video? We're going to talk about cell cycle checkpoints and what they are, what their role is in regulating the cell cycle, how proteins are involved in these checkpoints, and what happens if the cell cycle gets disrupted. So the cell cycle is regulated by these internal controls or checkpoints that check to make sure that the progression through the cell cycle is occurring correctly. You can see in our diagram that those are represented by stop signs. The first one here is called the G1 checkpoint. So at the end of the G1 phase, the cell is checking to make sure that it is the correct size, that it has all the correct nutrients and growth factors to progress, and that there's not any damages to the DNA. The next checkpoint here is the G2 checkpoint. So at the end of G2, the DNA is replicated, and so the cell is checking to make sure that that replication occurred properly, and it's checking to make sure, again, that there's not any damages to the DNA. Our last checkpoint is the M spindle checkpoint. At this checkpoint, the cell is making sure that the fibers have attached to the chromosomes properly so that mitosis can occur accurately and we can get the right amount of DNA into both of the new daughter cells. So there are interactions between things called cyclins and cyclin-dependent kinases that help to control this cell cycle. Cyclins are a group of related proteins that are associated with specific phases of the cell cycle. And so there are different cyclins that are involved in different stages. And their concentrations fluctuate depending on the cell's activity. So their amounts will vary depending on what phase the cell is in. The cyclins are produced when we need to promote cell cycle progression on to the next phase, and they can be degraded to inhibit the cell cycle from progressing. These cyclins are used to activate what we call cyclin-dependent kinases, or CDKs. And the cyclins are very specific to the CDK that they can activate. So those CDKs, or cyclin-dependent kinases, as you notice, end in ACE. So they are a group of enzymes, again, involved in regulating the cell cycle. They require that very specific cyclin to bind to them to be activated. And when they're activated, they can phosphorylate substrates and promote certain cell cycle activities to occur. Sometimes there can be disruptions to the cell cycle that could result in cancer or what we call apoptosis, which is programmed cell death. So we look at this first image, we see a normal cell that's going through the cell cycle, and the daughter cell from the first round of cell division here at the top has gone through some sort of genetic alteration. And so to prevent making copies of a cell that has a genetic alteration that could be harmful, the cell is triggered to go through what we call apoptosis and have programmed cell death. And you can see that happening in the diagram. Another possibility to a disruption in the cell cycle is seen here in this image. We can see the similar process occurring with the cell going through the cell cycle and making daughter cells. And the top daughter cell, again, has a genetic alteration. And this time it is able to skip past the checkpoints and continue to divide. And that division is able to continue to occur and have a buildup of cells over time, which can cause cancer. So let's look at an example. Here we're looking at, again, the interactions between cyclins and cyclin-dependent kinases, and specifically we're looking at one of the ones that is responsible for promoting M phase or mitosis to occur. So you can see that as the cell cycle is continuing, the cyclin that is responsible for promoting M phase builds up over time, so it's getting more and more concentrated. And as that cyclin is more concentrated, it can bind to its proper CDK, forming a cyclin CDK complex. This particular complex is called MPF, or mitosis promoting factor, because this is the one that's specific to promoting mitosis. It will then be able to do cellular work that is involved in promoting mitosis, and afterward the cyclin will be degraded, and then the cycle will start over where it starts to build up over time, make the complex when it's time for M phase, degrade, and onward. If we look over here at the graph, we can see that it is showing us that similar process, but we're looking at the relative concentration at the different parts of the cell cycle. So this red line is showing us our cyclin, and that cyclin is building up as we approach M phase. And then when it has peaked, you can see that the mitosis promoting factor also peaks in activity. We're able to accomplish the tasks that are associated with that phase, and then the cyclin will degrade until it builds up for the next round. All right, here's another example. This one is looking at a disruption to the cell cycle that could cause cancer. So we know that the original code for making proteins is stored in DNA. So it's possible that if the DNA code for a protein is altered, then the structure and the function of that protein could also be altered. So if there's a change in the DNA region that codes for one of the proteins needed that regulates the cell cycle, then it's possible that that cell cycle could go unregulated because that protein is not functioning properly. And so cancer would be the result of an unregulated cell cycle with uncontrolled cell division. So let's practice. The lesson skill focus for this video is argumentation, specifically making a prediction. So we're looking to predict the causes or effects of a change in or disruption to one or more components of a biological system. So here is our practice question. So go ahead and pause your video and give it a try. And then when you're ready, press play and we will discuss it. All right, so the disruption that this question is asking you about is that there was a mutation in the CDC15 gene that causes cell cycle arrest in telophase. So cell cycle arrest means that the cell has been stopped in that phase. So now we need to predict what is going to happen if the cell has stopped in telophase. And the correct answer choice here is answer choice C. The yeast cells will replicate their chromosomes, but will fail to complete cytokinesis. We know this is correct because replication of chromosomes happens during interphase, which is before telophase. And so that would have already occurred if we've made it all the way to telophase. But cytokinesis happens after telophase, and so we would fail to complete that step if the cell has stopped in telophase. So what should we take away from this video? Our key takeaways are that there are checkpoints that are regulatory events in the cell cycle. These checkpoints help determine whether the cell is ready to progress through the cell cycle. And proteins are used to activate or inhibit these cell cycle activities. Apoptosis and/or cancer can occur when the cell cycle is disrupted. Thank you for joining AP Daily, and I will see you next time.